point, which is, although in the past, doing this little intro talk, I've always been completely comfortable giving this talk for free. But this year, I have to say I'm a little disappointed that I have not yet noticed a deposit of $99,980 into my LLC account. That has not yet happened. So those of you in the room who are from Novartis, I'm assuming this is just some sort of clerical oversight. So if you could go check on that, I'd appreciate it. Because instead of the headlines that we saw last week, wouldn't we all have been much, much happier if this had been the headline that we woke up to? I know I personally would have preferred to see this headline than the one that I saw. So moving on, in our hands at Relay, FEP is working really quite well in cases where the problem is well defined. So there are two programs that we've been working on, and I'd like to credit the folks from Schrodinger. Relay has been collaborating with Schrodinger since the very beginning of Relay, a little bit over 2 years ago. So on this first program, it's mostly Satish and Eric at Schrodinger working with Pat and Levi. And by the way, just one minor correction, although it is true that I have over the years been an advisor to Schrodinger. I actually am no longer an advisor to Schrodinger, just for clarity, I wanted to make that point. But Relay certainly has been working very closely with Schrodinger on this program, and you can see that 60% of the time the results are correct within a factor of 5. And you notice there's a lot of points on this graph. It's more than 300 compounds that have been made in response to FEP calculations, something like 10,000 FEP calcs. I mean, Satish would know the precise number, but it's something on the order of 10,000 FEP calcs that have been run on this one program. And we've learned a lot from this process, and it gets better all the time. It's especially good when you're trying to explore broadly within a particular chemotype. It's also starting to work really pretty well for scaffold morphing as well. That's getting there too. So then there's a second program, and on this one it's mostly Goran and Fiona from Schrodinger that have been working with Pat and Levi and Dimitri. And again, the results are accurate within a factor of 5 to about 60% of the time. So we're really pretty excited about this because what it means is that when you have a checklist, when you know what the problem is you're trying to solve, you can with some confidence apply this technology quite broadly. And so this has got me thinking about how do we take this to the next step. So how do we go very broad with FEP? And when I say that, what I mean is, can we run hundreds or perhaps even thousands of FEP calculations for each and every molecule that we choose to synthesize? And I think that's the right question to ask, both because of the improvements in the quality of the results from FEP over the last few years, but also because synthesis is getting a lot better. So those of you who are familiar with how folks in the lab operate, there have been all kinds of advances in the last few years. It's really quite impressive. Now, the crotchety old chemists in the room, and I guess I'm one of them, could say, well, you know, it's still limited. The molecules you make are not that complex. The throughput's not that good, et cetera. But it's gotten so much better so rapidly, and the trajectory is so positive. I think we have to anticipate that synthetic capabilities will continue to dramatically improve. And so I think the right question to ask is it now possible to go much more broadly with FEP, and when is that the right choice? So I think that, of course, one obvious thing to get out of the way, first and foremost, is to do this, you would need tons of computers. But I'm totally with Asimov on this. I think the issue is if we didn't have enough computers, we should be scared. But I'm actually not too worried about this because of the tremendous progress that's been made and obviously will continue to be made. So sure, I wish computers were 10 times faster or 100 times faster, but they'll get there. So I think we can anticipate that problem won't hold us back too much. The bigger issue, I think, is our mindset. So we can't approach this problem entirely from a theoretical perspective. I love Kevin Kelly's phrase, thinkism. You can't think your way into a drug candidate. You can't cure cancer by thinking about it. We need to do experiments. And so the question then becomes, what are the right experiments to do to enable us to move faster? And so then, how do we dovetail the experimental capabilities with FEP or with any computational method? And so the issue now, as I think about it, it's gotten past sort of vague theoretical questions. It now is a matter of how do we put FEP into practice? How do we execute on the concept of FEP? And so for me, the whole issue here now is about the interplay, the balance between experiment and theory. How do we get that right to optimize our ability to use technologies like FEP to discover new medicines? So let's drill into this a little bit. We can think about it either from the calculational or from the experimental standpoint. So let's start with thinking about what FEP costs. Now, obviously, as I mentioned, Relay is collaborating extensively with Schrodinger on these programs, and so the vast majority of the FEP calculations are being run by Schrodinger. But just as a benchmark, if we try to run at Relay ourselves calculations, what do they cost? It turns out it's about $25 a compound. And you'll see that the hardware cost at this point is already a very minor component of that. And the major cost comes from the cost of the software itself, but also the human time. And so if you want that number to come down, you've got to work on those things rather than the hardware. Well, obviously, as the hardware gets faster, that brings down the licensing cost. And then as we get smarter about thinking about how to set up the calculations and to run them and analyze them, that reduces the human cost. And so my total guess about where we will be 2 years from today is $5 a compound. Now, obviously, many folks in the room are thinking about ways to bring that number down even further. So some folks in the room are thinking about whether there are, even opportunities for some kind of a lower cost or free implementation of FEP. I personally don't think that will be ready in 2 years. And so I'm not myself thinking about that, although I recognize that's an interesting and important topic. And the other thing that many folks in the room are thinking about are kind of pre-FEP, mini, smaller, faster, approximate ways of running FEP, which could further reduce the cost. But I'm ignoring the things in the blue box. And I'm simply saying, let's assume the price does gradually come down over the next couple years to about $5 a compound. So how would that affect the way we might think about deploying the method and using it? So on the flip side of the coin, what does it cost to make compounds? Well, if you go out to a Far East vendor, typically you'll pay anywhere from $1,000 to $5,000 a compound. Now, yes, of course, there's a huge variability. It depends on so many different specific aspects of the problem you're working on. What if you've got 50 grams of a key intermediate and you want to bang out a library? Obviously it'll be much cheaper. What if you only want 1 milligram of each compound? What if the chemistry is hard, low yield, steppy? And then you have all the stuff that happens after you've made the compound, all the assays, all the testing, which can be quite expensive. If you're running primary neuronal co-culture assays in low throughput, you're going to spend a lot of money on the assays. So you have to think about all of those things. But I think what's really interesting for us to contemplate is that the baseline for the cost of synthesis is definitely coming down. As I think everybody knows, it's happening in two different ways. It's happening at the low end in the sense that there are now vendors and even drug companies, I'm sure Novartis is doing some of this, a lot of companies are, these semi-virtual libraries where you have hundreds of millions or possibly billions of compounds that you're pretty sure you can make. And so you pull from that semi-virtual library and you go off and you make those compounds. Your chances of getting them are pretty high. Depending on who you talk to, you hear anywhere from 70 to 90% typically. The cost from some of these vendors is in the $100 range. They'll tell you they can produce them in 3 weeks. I'm seeing more like 4 to 6 weeks, but those things will all continue to improve, right? So that's very good at the low end. Now at the high end, the advances in synthetic technology, largely coming from a wide variety of academic labs, are just truly staggering. And so our ability to access very challenging molecules that a med chem team in industry, even 5 years ago, might have been very hesitant to go after. Our ability to do that now is much, much greater. Now, obviously, for those difficult to make molecules, you have to have a pretty high confidence in your hypothesis, or you won't go through that work. But it's at least a possibility now to contemplate making those kinds of challenging molecules. So whether you look at it from the low end or the high end, the progress that's being made in synthesis is truly astounding. And similar things, of course, are happening with respect to all kinds of assay technologies. And so in light of the ever-reduced cost of FEP, the improvement in accuracy, and the improvements in synthesis and testing, in light of the rapid change in all of those dimensions, then how do we want to think about deploying FEP? So I think that to really understand this problem, you have to look at it in the context of specific scenarios. You can't look at it in some abstract sense. And so I could throw out any number of possible scenarios. I'll just list a few. So what if today someone wanted to run 1,000 calculations at a cost of $25 each in order to pick one molecule for synthesis? So in that scenario, you're spending 25 times more money on the calculation than you are on the experiment. So how good would you expect FEP to need to be in order for that to be the right relative expenditure on theory versus experiment? Or imagine it's a library situation where you can bang out the compounds more quickly and cheaply. Maybe you can make a library of 50 compounds at $200 a pop. But you still want to analyze 1,000 ideas at a cost of $25 a piece. Is that a reasonable way to go? What level of accuracy would you need? How would you think about picking the 50? Is it based only on potency? Is it based on some multi-parameter process? What fraction of the 50 would you pick explicitly because FEP said they wouldn't work? How would you think about all of that? Very complex problem. Now, a third situation, very different, is when you're at the beginning of a program and you don't have a lot of crystal structures yet. You maybe don't quite have the same level of confidence in your docking poses. You don't have as many edges, and so the accuracy of your calculations is lower. Maybe you want to bias your thinking more towards experiment, generating more X-ray data. Maybe the best thing you can do is to make the most diverse compounds possible and generate as many crystal structures as you can. Do that for the first 3 months and then start applying FEP, for example. That might be how you'd think about it. So each of these are specific scenarios, and so the way I'm suggesting we come at it is to look at each case individually and then ask, what is the optimal blend of experimentation and theory to get us where we need to go? So I say break out the green eye shades. I say that although all of us as scientists and engineers are used to thinking big, I think in some sense we have to go very deep and down into the weeds and really understand in minute detail how to apply the technology. And that's a very case-by-case, very specific kind of a situation to be in. So that's my suggestion. Now, I think if you then accept this idea that every situation is unique, here we are in Cambridge, the hub of the universe. By the way, for those of you who don't live here, welcome to the hub of the universe. And so if you go over to Sloan, I love teaching at MIT, and you go over to Sloan, you talk to the systems engineering guys about problems like this, and you say, well, here's what I'm attempting to do. How would you think about it? And they'll start asking you all kinds of very, very detailed questions. They'll say, well, what exactly is the problem that you're trying to solve? Are you trying to optimize for CYP binding? Are you looking for selectivity, chemical diversity? What is it you're doing? How accurate do you think the results are, and why do you believe that? How interested is the team, the synthetic team, in pursuing the compounds that you're talking about running calculations on? And that relates to what's the turnaround time that you need to achieve in order to have an impact on the thinking of the team. What will it cost them then to make all the compounds you're proposing? And conversely, what will it cost to run all the calculations that you need to run to make the predictions to then make the suggestions? Are you looking for an order of magnitude kind of answer because you're exploring broadly new chemical space and you're looking for stakes in the ground? Or are you trying to decide where to put the fluorine in order to optimize your drug candidate? And if you didn't have access to the technology that you're discussing, in this case FEP, what would you do? How would you make your decisions? Would they be less accurate? Why do you think so? Under what circumstances? So these are the kinds of questions that a systems engineer would ask you about how you would think about deploying a technology like this. Now, I would argue that most of us in the room are not used to thinking that way. It's not that we're incapable of it, but I don't think it's what we naturally do. I think we tend to think about the science in more basic terms. But my argument would be that if we're trying to understand now how to use this technology in the optimal way, there's value in going down into the weeds. And I also think that from a basic science standpoint, this helps us because it helps us to identify which are the gaps in the technology that are the most important to fill. What are the fundamental challenges that remain that if only we could tackle them, they would have the biggest impact on the accuracy or the sphere of applicability or the throughput of the method. And this afternoon session will get into a lot of that. So I think it's important to really have to have people on the team who really do think like system engineers or even think like accountants. It's like in Archer. If you're going to have an accountant, you want a really darn good accountant on your team. For those of you who are Archer fans. So, by the way, this is just like every other technology in history. Whether it's high throughput screening, second harmonic generation, and analysis of synergistic effects from combination studies in 1,000 cancer cell lines, DNA encoded libraries, whatever it is that you're talking about, cryo-EM, you have to ask these questions. What will it cost to implement and validate and deploy this technology, and what benefit does it provide, and why is it better to do this than that? Why are you better off if you buy a Biodyssey at $500,000 and you're paying $2.50 per well because the darn plates are $800? Why is that a good expenditure? What do you get from that? Why is that cost effective? Right? You have to ask those kinds of very precise questions. So this is good news, because what this means is, in my opinion at least, FEP now is accurate enough that we should be taking it this seriously. We should be asking this level of detailed questions about how to deploy it, how to operationalize it, which I don't think was necessarily true 5 years ago. So that's good news. So you may have picked up by now that I'm suggesting that we focus on operational thinking. I hope that's come through. If it didn't, I could go back to the beginning and go through all the slides again. But how do we use that insight, assuming that you think it's an insight worth pursuing at all, which you may or may not? What would we do with that? Where does that lead us? Okay, so I would say it gets us thinking then about, as I mentioned, the nature of the problems that we're trying to solve and what would help us to do better at cracking those problems. And I think fundamentally it always comes down to three buckets. What does it cost? How accurate is it? And to which problems does it apply? And so for in each of those three buckets, you can define any number of subtasks, subtopics. And I'm not intending to be exhaustive here. I'm just listing some of the ones that I know are of interest to different folks in the room, and I'm sure I've left a few out. And I know some of these could go in different buckets or multiple buckets. It's not always as clean as what I'm saying here. But the basic point is there's a lot of choices that we can make. And what'll be great, I think, is to hear the progress that's being made in each of these different areas. And I know a lot of this will be discussed at the meeting. So just to talk about one of these, because personally I'm very interested in throughput. Very interested in this idea of being able to scale this up and go much, much faster than before. And so the idea of prefilters is particularly relevant, at least to the way I'm thinking about this problem. So when one thinks about prefilters, I think there's really three different categories that you can imagine. One is related to intrinsic, affinity, binding potency. And that could be either on target or off target. The second is the breadth of the chemical space you're exploring, how much diversity. And then finally, it's the broad bucket of properties, whether it's physical or DMPK tox properties. All three of these are important. So if you have a molecule that's 10 times more potent, maybe you can lower your cellular dose, which can be incredibly useful for drug design. Obviously, a more selective compound may lower a risk of a side effect. More diversity, different chemotypes may have fundamentally different properties, may get around a PK problem that you can't otherwise get around. And of course, as we all know, properties are the big issue. And this is really critical now, because I think with FEP, what we're seeing is we can bottom out the biochemical assay on these problems that I described before. So on the first project, the one where we've run 10,000 FEP calculations, we've now got something like 15 different chemotypes that we've patented already, and there's more coming. And so the breadth of chemical space we've been able to explore and really drive the potency down to, to the bottom of the assay is really spectacular. And so that is no longer the issue on that project. The issue is if every one of those chemical series requires you then to scale up the compounds and take them into animal studies, it's still going to be a very slow and very expensive process. And so what you really need to do is figure out how to weed compounds out prior to those late stage, end game, lead up kinds of assays. And so that, I think, is where the game now needs to be played. So for me, Prefilters really is all about reducing the burden in the end game. And so that's how we're coming at the problem. So just a couple of examples of Prefilters where we're using machine learning. The first is from a frequently annoying DMPK tox problem. It's a parameter that everybody worries about quite often. Doesn't matter which one it is, actually. But you can see that using about 200 experimental data points and some other information, we've been able to build a machine learning model that's pretty good and enables us to throw away probably 3/4 of the compounds that otherwise the chemist would be thinking about synthesizing. And also, we're thinking quite hard about our ability to predict binding itself using this sort of machine learning approach, because we're very lucky at Relay, because of our collaboration with David Shaw's team. We have access to probably 1,000 times more compute power than literally the rest of the industry combined. And so the question is, what can we do with that? Is it good for anything? I think that's an open question. And so, I'm not presuming that it is. So the question we're asking ourselves all the time is, well, with that ability to run, hundreds of microseconds of simulations on each compound, what does that do for you? And so we've been asking, how do we use all of our biophysics data, all of our structural data, all of that simulation data? How do we smush that together with some, we hope, intelligent molecular representation, which is not a solved problem? How do we use that in some sort of machine learning approach to build a predictive model on binding affinity? And it's working okay. Probably got an RMSD of about a kcal.5. So that enables us to throw away maybe half of the ideas. And so imagine that we have many of these machine learning models that we're building, and we'll obviously, it's work in progress. We hope that we'll keep getting better at it. But the idea is to use all of these as prefilters, so the number of compounds that needs to go into FEP is as small as it possibly can be. So in closing, by the way, this is work that's been done by Molly and Pat and Levi at Relay, and we've been working with the Reverie folks on this. Some of you, I'm sure, know them. They're very good. So we're very happy with the relationship we have with Reverie. So in closing, we know there are many exciting and interesting problems to work on to help make this technology better. Many of you in the room are already working on these. A lot of the discussion this week will be about the progress that's being made or the plans that you have to work on these different topics. You can debate endlessly, which of these are the easiest to solve, which of them will have the greatest impact. My argument, as I've said this morning, is we should think very carefully, as quantitatively and precisely as we can about that to help us focus on the things that matter the most. Some problems may be a little too hard. I'm suggesting at the bottom, the ones in yellow, you could disagree with that. Some problems are intrinsically easier than others, more approachable given what we know, given the amount of compute power we have and so forth. But whichever ones we work on, it'll be great, obviously, at this meeting and also at the meeting 2 years from now to hear about the progress that's being made. And so the last thing I'd like to say is I'd like to ask the organizers if they would be comfortable taking on one additional responsibility. I think that what we should do this week is we should award at the end of the meeting. We should give an award to whichever speaker. And it's not me. Let's be clear. Whichever speaker does the best job of helping us to think quantitatively about the benefit of FEP in a specific circumstance. And I would propose that that individual be awarded the inaugural green eye shade. And this could be a cherished heirloom, which would be passed down from meeting to meeting. So would the organizers be willing to accept this sacred responsibility? Okay, well, here, the torch has been passed. Okay. So that's it. Hope this was interesting. Feel free to ask questions or we can discuss later. Thank you. Yeah.
Just a simple question about your cost estimates. Were you including, did that include like $5 per FEP per compound? Did you include in that the cost of the technician or the scientist who runs the calculation?
Yeah. So the three components are the hardware, the cost of the software, and the human cost. Okay. So the human cost would be all the time that a scientist needs to take to.
Right.
Yeah.
So is there any force field development in that cost?
No. No, because we're paying, in that model, we're paying the license fee for the product. But you're right. So it's a very good point. In addition, if we were doing our own basic research to further extend the technology, then yes, that would be an added human cost for sure. But that wasn't part of that calculation.
So you said the free versions aren't there. You don't think they'll be there for 2 years. What are the things holding it back? The force field, the software design, something else?
Well, first of all, I appreciate the fact that you're asking me that question like I know better than like everybody else in the room, so thank you for that compliment. I actually don't know that I can give the best answer to that question versus everybody else here, but I think it's a little of all of the above, right? I think it's ease of use, I think it's robustness, it's speed, it's force field, it's sampling. I think it's all of the above, right? So that's why for me, because the cost is going to come down to $5 over the next couple years, that's why I think it's not, for me personally, the most important thing, whether the software is free. The more important thing is what I talked about, which is how do we want to use the technology in the context of drug discovery versus all of the other choices that we have. That's not to say it isn't an important topic, just not for me, it's not the most important topic. But I do think it's a complex problem. You know, I think we have to recognize this is an old field. It's been around forever, and many people have put their whole careers, many people in this room, you know, have put a lifetime into studying this problem, so it's not going to be trivial to just, you know, crank out something that's better. At least that's how it looks to me, but other folks in the room will have other opinions about that, I recognize. Yeah?
Mark, I love your packaging of all other computational methods into pre-FEP. Most of your talk was about it. It's not about the FEP, it's about the drug discovery. Do you see any of the methods emerging as post-FEP?
Okay, well, first of all, I'm glad you asked the question because it means that I did a very poor job in my talk, because what I was really trying to get at was the idea that FEP is a part of the whole process of drug discovery. And as I said, it's working really well, so we want to do more and more and more FEP. But in order to enable that, we have to think about it in the broader context. And so I wasn't in any way trying to say we don't need FEP. Okay. So as far as post-processing, I think that, you know, there's two things that are attractive to me. One is the ability to analyze the trajectories. You know, if you're running thousands and thousands of simulations, it becomes kind of a difficult task to analyze the motion of the proteins and the ligands and the waters, you know, over the time course of the simulations. So I think there's going to need to be a lot of work done to try to automate that process using a wide variety of approaches, a lot of machine learning and other technologies. A lot of that work already is underway, obviously. That's one kind of post-processing. Another kind of post-processing is, you know, I've not quite given up yet on the idea that we may someday be able to have a more human interpretable conclusion from the FEP calculations. This is actually something that Robert and I have talked about quite a bit. And Robert so far has convinced me that we don't really know how to do that. And I think you're right, Robert, that we don't. And you may, Robert, you might actually think it can't be done for fundamental reasons that you understand better than I do. But I'm still, as an end user, as a chemist, I'm still hoping that eventually we will have ways of analyzing collections of results from FEP that then can be interpreted in some human way to try to guide thinking. That may be foolish, that may be 20th century thinking, I recognize. But you asked about post-processing, so those are the two things that are on my mind. The analysis, the interpretation of all the trajectories, and whether there's some way to interpret, in a human way where the energetics are coming from. Yeah.
Okay, well, let's thank Mark again for a very nice kickoff.